Dr. Lazarus holds a PhD in marine sciences, and has over 15 years of experience in marine ecology, conservation, and restoration. He has contributed to multiple research projects for the declaration and evaluation of marine protected areas in Colombia, and has led coral reef restoration initiatives on the Pacific coast. Today, he will present planning, implementation, and monitoring of mangrove recovery, a case study of best practices in the Ciénaga Grande of Santa Marta. We welcome Dr. Lazarus.
Buenos días a todos y todas.
Good morning, everyone. I'm pleased to be here. I'm Juan Felipe Lazarus. I belong to Invemar, and I lead the team for marine and coastal recovery of ecosystems. Today, we want to tell you a bit about recovery restoration process that we have started in the Ciénaga Grande of Santa Marta. In this case, this is an approach where we integrated different disciplines, and we do a holistic job on the ecosystem, which we have called planning, implementing, and monitoring mangrove recovery.
From Ciénaga Grande. Listo. Bueno, como todos entonces sabemos,
as you all know, mangrove are very important blue carbon wetland ecosystem where we find this capacity for carbon sequestration. And the storage in this plays a crucial role where we have this climate biodiversity crisis. And of course, this is a call to action, and this call to action we've made it real through different agreements and global goals and commitments, such as the Sustainable Development Goals, the Global Biodiversity Framework, the goals of the Oceanic Sciences, and the Decade of Restoration led by the United Nations. Here we have a very interesting ecosystem, which is the Ciénaga Grande of Santa Marta. As for those who know, this is our laboratory. We have done a vast amount of work in this area concerning the recovery of this very important coastal wetland, and this is the biggest coastal lake, a Ramsar site, and a international biosphere reserve, and it's got different protections at the national level. And there we have the ecological and ecosystem services importance, which is, this is a hotspot for the biodiversity and a carbon storage site. And there is also importance in fisheries. This is 6.5 thousand tons, and this is food security for 3,500 fishermen and their families. And it's been an ecosystem that has gone through a very significant events, well, catastrophic events, better said, that has caused degradation, construction of two roads, one first road connecting Ciénaga with the city of Barranquilla, and another one that's probably to one of the biggest rivers in the country. And these produced a water misbalance by the 50s and the 70s. This generated a loss of 60% of the mangrove coverage of the area, and this is the result of hypersalinity, sedimentation, and canal blockages. So, and this compromised carbon sequestration capacity, and we need an urgency to restore these ecosystems. Here we have that from our position, we found that it was important to have a baseline that was robust enough from the ecological point of view, hydrological point of view, and we then devoted to that. So we made an assessment to establish a baseline so we could have the technical criteria to conduct the restoration, but also together with the communities. So there we created these channels for the communities where we found community groups that were interested in the process, who in addition integrated not from the technical perspective of the restoration and ecological perspective, but also by strengthening their administrative capacities so that they can lead their own processes and observe conservation and preservation and have it as a livelihood in the long term. And this was accompanied by a monitoring proposal so that we can follow up the restoration process in time, and also for decision making for the adaptive management of these actions, and also check if these actions reflect effectively on an improvement of the ecosystem quality and carbon sequestration. This is our area of work. This is 30 hectares in size. There we also have about 300 hectares in restoration processes, but in this case we focused on this polygon here. And here we identified, well, thanks to this baseline, precisely, we knew what we had to do in hydrological terms. So there we registered salinity, water levels, and water flows and dynamics. That also allowed us to identify the water bodies and flows that we had to intervene in order to achieve an improvement in water conditions. From the ecological perspective, we worked in species density and also the health of these forests and how they changed in time. And in soil, we understood the type of sedimentation on site, sort of nutrients, and something important that is why we're here, which is the content of carbon in this soil. And something else that was crucial for our objective. What did we find? We found this main canal that we hadn't identified initially in our initial assessment that was totally blockaded. This then forced us to do something else. In addition to what we had planned, we had to intervene 3.5 kilometers that were totally obstructed by macrophytes so that we could guarantee the flow of water into the polygon. Once inside the polygon, we worked for around 2.5 kilometers of secondary channels, and then we started to observe an improvement in water, reduction in salinity year on year, and every restoration of the flows that allowed a washing of the soils. Together with the communities, we conducted these activities. We did some manual work, and we identified that some of the areas in our study site presented this permanent flooding, lost elevation, and salinity levels were pretty high. And this is in order to bring new plants. So we brought these dispersion monticles. Here we are able to elevate the substrate, so the plants found there are a bit higher than the flooding levels so that they can grow. We had 200 dispersion monticles for 2,000 samplings for white and black mangrove, and we established also this sediment piles so we could use that material in the area. And for that, we wanted to find new areas for natural restoration so that we could then settle the seeds for those saplings that were produced locally. So these are the plants, the monticles, with high seeding survival, with a growth of 80 centimeters in 5 months for the racemosa, and also the presence of new recruits in the area thanks to the natural regeneration. With the communities, always close, we started to design our actions. We started to adapt the plan that was established initially, sorting out those challenges day after day. So it was very common to have these dialogues in the field, making decisions jointly with the communities. Here, we achieved active participation of communities, where we also improved or strengthened these communities technically and administratively speaking. And this brought us to a commitment in the long term and the sustainability of this process, where community organizations and grassroots organizations independently were able to conduct maintenance of activities and monitoring activities. In relation to the results of the monitoring and water quality, which was one of the biggest problems, and thanks to the opening of these canals, we were able to reduce salinity in our intervention site versus our the site with no intervention. And we have these a benchmark forest, and between January 2023, the yellow bars, and February 2024, you see how on the same site there is improvement in conditions of about 10 points in salinity. We also see that we measured hydro periods in our sites, and we see that there are similar rhythms to the one in reference, and that means that the water is flowing, whereas the non-intervened site, we see that water is not flowing. Concerning ecosystems and ecology, at the Institute, we have an indicator of the status of a mangrove, telling us about the health, including different variables, basal area, EAP, DAP, species composition, and we also have the natural regeneration included in this index or indicator, including saplings and seedlings and salinity. Once we have all these values, then we proceed to calculate an index or indicator, and with that, which is important for decision makers, they understand how the mangrove is behaving. So this is the intervened mangrove going from a category of regular or into a good condition. There we see how these trees have increased their height and diameter, and we also see that there is a change in the population of these forests having mature bigger trees. Something important for us now that connects with carbon sequestration is that both hydrological rehabilitation and new planting create optimal environments for the sequestration and storage of carbon. There we see that we were able to achieve organic carbon content higher than the ones we observed initially. Also, that anoxic conditions changed, which guarantees the stability of this carbon in time. And we also saw how nutrient dynamics improved, including phosphorus, which guarantees the stability of the carbon retention. And here we see how, thanks to these actions, we obtained results and contribute actively to the removing of CO2 from the atmosphere storing it
carbono azul.
as blue carbon.
Aquí tenemos que incluimos y mejoramos
Here we included and
la
improved
cobertura de
the
manglar,
coverage
la
of a
diversidad
mangrove and
y
its
una
diversity
calidad del
too,
agua.
and improved water
Hay
quality too.
almacenamiento de carbono activo en el
There
área
is active
que se
carbon
refleja en todos
sequestration
los
and
ejercicios
storage in the
medidos en
area,
suelo con
reflected in the
datos
content
medidos
of the
y
soil
además
when we
la
measured
biomasa
them, and
ganada
also
con la
the
imagen
biomass
vegetal
that we
que se
gained
está
with the
estableciendo. Y ustedes
vegetation
pueden
that
ver
was
cómo
being established. And you can
de
see
febrero
how
de
from
2024
February
a octubre de 2024
2024 to
cambian
October
estos pequeños
2024,
islotes
these
porque buscamos
small
precisamente es
islets
que en el tiempo estos
changed.
islotes
So with
se
time,
encuentren y
this
formen una matriz o un núcleo
islets
de mangrove.
can form a matrix
Vemos también que hay una mejora
of mangrove. There is also
de los medios de vida de
improvement
las
of
comunidades,
the
donde
livelihoods of the
se
communities,
implementaron mejores
where
políticas de pesca
best practices for
y
fishing were
unas
implemented,
actividades de turismo,
and including some
de las cuales
ecotourism
quienes nos
activities.
acompañen el día jueves a campo
And
van a poder
those that
conocer a
are
una de estas
going to
personas
come with us on
que
Thursday are going to meet
ha
some of
hecho del
these
ecoturismo un
people
medio de
and
vida.
community members that have
Además,
made a
gracias
business
al
of
empoderamiento de
ecotourism.
las
And
comunidades,
also,
hemos logrado
thanks to
también
the
un
empowerment of communities,
manejo de
we
estos
have
importantes
achieved
ecosistemas.
effective management of these ecosystems.
Y aquí tenemos unas
And these
lecciones
are
aprendidas.
some of
Primero
lessons
es
learned. One of them
realmente
is
necesitamos tener un
that we
conocimiento
must have
y una
a
buena línea base.
the knowledge
Tenemos
and the
otro
proper
punto importante
baseline.
para nosotros
And
es que es muy importante
something else
lograr la
also
integración
important
del
is
conocimiento
that it's important
local
to integrate local knowledge and
científico y que esta
scientific
relación
knowledge,
sea una
and
relación
this
horizontal
relationship
y
must
no
be
siempre
horizontal
como suele
and
verse
not as
en las
it is
políticas. Y
habitually seen
finalmente
in a vertical fashion.
es
And
importante
finally, it is
lograr
important to achieve
la vinculación
the
y el commitment
engagement
comunal,
and
lo cual
participation
nos
of the
garantiza el
community
exitoso
so
la
that
sostenibilidad del
we can
proceso.
ensure the sustainability of the processes.
Finalmente,
Finally,
y como un
and
mensaje
as a final
o un último
takeaway,
mensaje, esta experiencia en la Ciénaga
this
Grande de Santa
experience
Marta
at, uh,
es más que una
at the Ciénaga
intervención,
Grande of Santa
es
Marta is more than
un proceso
intervention.
holístico
It is a holistic
que logra la integración del conocimiento
approach
local con el
that
científico
integrates local
y
knowledge with
aquí
science. And
el compromiso local
here,
entonces nos permite
commitment
recuperar
from the local
estos
allows us
importantes
to
ecosistemas de
recover,
carbono azul
restore these blue
que
carbon
además
ecosystems
contribuyen a la
that
resiliencia de las
also contribute
comunidades
to the
y nos permiten
resiliency of
avanzar en los
communities
retos
and allows us
globales
to advance
que
towards
tenemos
global
como
goals we
país.
have as a country.
Muchísimas
Thank
gracias.
you very much.
Bueno, muchísimas gracias al doctor Lazarus.
Thank you very much,
A
Dr. Lazarus.
continuación, vamos a
Up next,
escuchar
we're going
a la
to
doctora
listen to
Rahanna
Dr.
Juman,
Rahanna Juman,
subdirectora del
Deputy
Instituto de Estudios
Director of the
Marítimos
Maritime
de Trinidad y
and
Tobago,
Research
y
Center
también
of
a
Trinidad and
Hamish
Tobago, and
Asmath,
also Hamish
oficial de
Asmath.
Sistemas de Información
This is
Geográfica
official
del
for Geographical
instituto.
Systems in the
La
same
doctora
institute.
Juman tiene un doctorado
Dr.
en
Junman
zoología
has
con una
a doctorate in
licenciatura en
zoology,
Derecho
license
y
in
ha
law,
dedicado casi
and
3
has
décadas
dedicated
a la
more than
investigación de
3 decades in
ecosistemas
the
costeros,
research of
incluyendo manglares y praderas
coast
marinas.
ecosystems, including
Su
mangroves
trabajo ha
and
contribuido a
seagrass.
políticas como la
Their
gestión
work has
integrada de
contributed
zonas
to the
costeras
politics
y
and
la
the integral
planificación
management of
espacial marina,
coastal areas
y
and also
actualmente
maritime planning, and is now
proyectos
leading
de
projects
restauración y
of
carbono
restoration and
sostenibilidad
blue
del
carbon in
mar.
Trinidad and
Y
Tobago.
el señor Asmat
And
es
Mr.
candidato a
Sasmat is a
doctorado de
candidate to
geografía en la
Doctorate
Universidad
of
de las
Geography
Indias
in the
Occidentales
University of the
en
West Indies
San
in the
Agustín.
San Agustín
Su
campus.
investigación se
Their
centra en
research is
hidrología de
based
cuencas,
in
hidrodinamics
hydrology
marina
and
y el uso de herramientas
hydrodynamics and the usage of
geoespaciales
geospatial
avanzadas.
tools.
A continuación,
Up
vamos a
next, we're
escuchar
going to
su
listen to their
presentación que se
presentation,
titula
and the
Modificaciones
title is
antropogénicas en el
Genetic
pantano
Modifications
de
is the
Caroni,
Wetlands
Impactos
of
y Servicios
Caroni,
Ecosistémicos.
Impacts and Ecosystemic Services.
Adelante.
Good afternoon and Thank you for inviting us to participate in this very important dialogue. So we're talking about one of our largest mangrove forest in Trinidad and Tobago that has been modified over the years, some of the impacts, but it still continues to provide us with ecosystem services. So this is the Caroni Swamp. It's, as I mentioned, our largest mangrove forest, and it's within our Gulf of Paria coastline. That's the river, the sea that we share with Venezuela. And it's just about 10 minutes away from the major city of Port of Spain. So it's very close by. And it's the hydrological endpoint of the Caroni River Basin, which is over 87 hectares. This is just a close-up of the mangrove mapping. So 70% of all the mangrove on our west coast are found in the Caroni Swamp. It is our largest mangrove forest, but our second largest wetland. The largest wetland is both mangrove and freshwater marsh. Back in 2020, we estimated it over 4,000 hectares, which is big for a small island like Trinidad. We have in that particular wetland 190 bird species recorded, including 20 endangered species. And of course, it's home for commercially important species like oysters, mussel, conch crabs, and fish. And we have people that earn livelihoods harvesting these organisms. And it's also very important for ecotourism. It's one of our major ecotourism sites in Trinidad and Tobago. Now, this is the river basin that flows into the Caroni Swamp, and it's 18 rivers that flow into the swamp from the land side. We have two major dams within the river basin. It is within that river basin, we have 33% of the population who are living there. So it's quite a dense, it is the most densely populated part of Trinidad and Tobago, and you'll see the red running along. That's what we call the east-west corridor, where most of our population lives. We have a lot of industries in that area, as well as agricultural lands. It used to be a lot of sugarcane cultivation, agriculture within that river basin, as well as we have gravel quarries. And with that comes the sedimentation that comes in, as well as limestone quarries. So this is just an idea of what the river basin and all the activities that occur in the river basin, and then eventually everything's flow into the Caroni Swamp before it flows into the ocean. In terms of legal status, it was a forest reserve claimed back in 1936 while we were still a colony. And it was further expanded in 1953 because it's a very important habitat for the breeding of the scarlet ibis, which is the national bird of Trinidad and Tobago, as well as an environmentally sensitive species. And they keep on extending the sanctuary again to accommodate the bird harvest, the scarlet ibis. In 2005, we were able to have it declared a wetland of international importance or a Ramsar site. Now, this is just giving you an idea of how the hydrology in this wetland has changed over time. So pre-1920, this is what it looked like, the first image. And then during our colonial time, there was something called the Cipriani scheme, where they decided that they wanted to drain the wetland to plant rice. So they changed up the hydrology, created a number of drains and an embankment, the north-south drain, a drain and a big embankment to keep out salt water. And this area, the reclaimed area, was supposed to be for rice cultivation, and they created a number of channels. Back then, the intention was to drain the land. So we put in conduits to drain the land so people can live. Right? Of course, over time, in most Caribbean islands, we don't maintain anything, right? So a lot of the infrastructure that was put in initially has fallen up, has not been maintained. So a lot of the sluice gates and so on that was put in to keep out the salt water, those went into disrepair. And this is what it currently looks like, the last imagery. And this, of course, has impacted what has happened to the Caroni Swamp itself. So the first imagery is 1942, right? And you would see the dark green is the mangrove area, and the light brown is the marsh area where they were supposed to plant rice, where there was rice cultivation. But over time, as the infrastructure broke down, and you had salt water coming in, sea level rise, because the same conduits that they would have constructed to drain the land was now bringing in the seawater. You've had the mangrove expanding, and you see it this would be from 1942 to 2007, where the mangrove, the green has expanded, as well as urbanized areas or buildings, which is the red, right? Of course, when you put in highways, and we have constructed a major highway through the wetland, you would have had changes that have occurred. So this is one area where we actually have mangrove expanding landwards, but at the expense of freshwater marsh vegetation. And this, of course, has an impact on even the scarlet ibis, because they require freshwater. Young scarlet ibis requires freshwater invertebrates, and they're not getting that freshwater invertebrate. So what you find is that they're moving further and further inland by the highway, where they become more vulnerable to poaching and so forth. And this is just a current, 2014 land coverage of the Caroni Swamp, again showing the mangroves. We are now trying to update that with more current imagery that we recently got through another project. What is also interesting is that within the mangrove too, that light area is actually area where, or bare areas area where we are noticing dieback. Within the center, the swamp, areas that are not as accessible. And we suspect a lot of it has to do with the change in the hydrology and some of these areas not being flushed as it would have happened in the past. Right? Along the coastline as well, you will see from 1957 to 2014, we've lost about 20 hectares of mangroves along the coastline. So while we have them growing inland and expanding, we are losing along the coast. And again, this is due to sea level rise. These through a project that was done by the European Union, we were able to do some simulation using SimClim and looked at actual sea level rise value for RCP 4.5, and this was for 2050 and 2100. And based on these values, we were able to show the level of flooding, right, with sea level rise. 2050, that's the 48 centimeter, and 2100, which is, you will see most of the swamp would be inundated. So again, in terms of coastal flooding and its role in terms of coastal flooding as well. Back in 2019, 2020, we also did some work where we were looking at pollution entering the mangrove swamp and trying to determine the source of the pollution. So we did isotopic analysis. We worked with Professor Valiella out of the Marine Biological Lab, and we looked at isotopes, nitrogen isotopes, to link it to what was the source of the pollutant. On the northern part of the wetland, which is the Caroni River itself, the main source of the pollutant was determined to be wastewater from sewage. However, on the southern side of the wetland, the main source of nitrogen pollution into the wetland was actually from agriculture, from farming. And we then set up a project to look at alternatives to using fertilizers, so the farmers can continue to grow their crops but reducing their pollution. What we also found out is that the swamp, again, plays an important role in terms of water quality maintenance, because when we looked at what entered and what eventually went through the wetland and comes out into the Gulf of Paria, we saw that 20% of the ammonia, there was reduction by 20% in terms of ammonia, 41% in terms of nitrate, 23% in terms of dissolved organic nitrogen, and about 25% in total dissolved nitrogen. So again, a lot of that is being absorbed by the Caroni, the mangrove system, and it's reducing before it goes out into the Gulf of Paria. So that's another ecosystem service. So in the past, we would have done biomass measurement, mangrove biomass measurement, using allometric equation, the typical ones that we would have used over the years, Golly et al., Sintra Novelli. But in 2018, we started to, we got 2014, we got LiDAR imagery. And in 2018, we started to quantify the amount of carbon in the above ground biomass in the mangrove forest, not only in Caroni Swamp, but throughout Trinidad and Tobago. And using the new methodology that the Terra laser scan. And since that time, we have continued to do work. We currently have an IDBTC that we're executing in terms of developing a blue carbon credit scheme for Trinidad and Tobago with the focus on Tobago, but comparing it to the Caroni Swamp, data collected for the Caroni Swamp. And we also have another project we're doing that is funded by the Green Fund, and it's being executed by the Climate Change Caribbean Community Climate Change Center. So I'm going to hand over now to my colleague, and he will tell you some more of the work that we'll be doing with these projects, at least with the Green GCF project.
Good afternoon, everyone. So with regards to LiDAR, us at the IMA sort of kind of fell into blue carbon sort of accidentally almost. In 2014, the government of Trinidad and Tobago commissioned a LiDAR survey for the whole country. And so we got the data in 2016 and processed it. So we got elevations of the ground and we got the tops of all the trees. So then I just thought, let's start. We're doing another project in Caroni Swamp. So I just started measuring trees. And lo and behold, 7 years later, we're doing all this other work with all you great people. So we measured throughout the country, we actually measured about 26 plots, 14 of which were actually in Caroni. And using the regular allometric equations, which we pulled from the region, we were able to get the plot level biomass. And then taking wood cores, we got the total amount of carbon in the trees and able to get plot level carbon and correlate that very, very strongly with LiDAR top of canopy height. So we had a R squared of about 0.88. So very good agreement. So we were able to come up with the assessment of mangrove carbon throughout Trinidad and Tobago. In Caroni Swamp, as you see here, there was roughly 573,000 tons. For all of Trinidad and Tobago, the mangrove carbon in the above ground was 876,000 tons. We also did a comparison between the per hectare coverage, and mangroves were 122 hectares as opposed to tropical rainforests of Trinidad, which were around 77 tons per hectare. So as we all know, mangroves hold more carbon, but it was good to actually quantify this. And a lot of the decision makers and policymakers in Trinidad just don't really know and appreciate this. So it was good to definitively demonstrate this based on local data. And I should say that forgot to mention that Caroni Swamp holds 65% of our above ground carbon biomass in Trinidad and Tobago. So as Rahanna said, we did that stuff basically up until 2018 and then into 2019. The Green Climate Fund, we got funding from the Green Climate Fund to do an update basically. So we're currently in the process of measuring, again, reassessing our plots from 2018. And these are the number of plots we're doing throughout Trinidad and Tobago. So it'll be a total of 52 plots, of 43 in Trinidad and 9 in Tobago. And so currently we have a LiDAR drone, which is the image on the left, and we will be using that to survey certain areas of the swamp. Drones are not possible to survey all of the mangrove areas of Trinidad and Tobago. So what we're going to do is we're hoping to get access to TandemX data, which will be able to use the LiDAR drone to validate the top of canopy height and be able to effectively assess at the landscape level. I personally prefer LiDAR as opposed to drone imagery, as Steve was talking about earlier, but that's more of a preference just because of what I did on my experience. And so these are some results of these plots that have been resurveyed from 2018. The light blue bars are the plots in 2018, and the dark green bars are the plots in 2025. So as you could see, most of the time there's been a significant increase in the amount of above ground carbon in tons that's been stored. The plots that have experienced a decrease, this is simply because trees fell over and died. So this is just the living above ground carbon. We of course assess the biomass on the ground, the down carbon, but we just didn't include it into the this process as yet. And of course, that eventually goes into the soil carbon. And so moving on to soil carbon, we got a small grant from the UK High Commission in Trinidad and Tobago in 2021 to conduct a pilot project assessing soil carbon for Trinidad, which has never ever been done before. So we didn't have enough money to buy cores. We built some cores, and this was actually a regional project between Trinidad and Tobago, Suriname, and Guyana. And the cores we were able to get in Trinidad went down to just over a meter, and we were able to test the carbon. And the result was, roughly speak, 18 tons per plot, which worked out to be 433 tons per hectare. Again, this is only 5 cores. But if you remember, the above ground carbon where we got for mangroves, the average for Trinidad and Tobago was 122 tons per hectare, and below ground carbon was 433. So as expected, and as we can know, a lot more. So we'll be doing a large number of cores across Trinidad and Tobago, approximately 118 cores, to come up with a much more comprehensive assessment of mangrove soil carbon for Trinidad and Tobago as part of this GCF project. Now, carbon storage is carbon storage is how much we have there. And of course, if you do a carbon storage assessment from 2018 to 2025, you'll know how much you've increased over the years. However, we'd also like to see how carbon storage sequestration occurs on an annual basis. So as part of the project, we've been able to get, we will be getting a leaf level photosynthesis analyzer. So we'll be able to see the carbon assimilation in real time. Pairing, this is an idea which I'll be happy if people could critique, because we have now putting it together. Or we'll use stand level leaf area index measurements, as well as satellite leaf area index measurements, to extrapolate from measuring this much of a leaf through the vertical profile of the mangrove tree, and then extrapolating from this to the leaf area of the entire mangrove canopy in Trinidad and Tobago, in conjunction with incoming solar radiation measurements, because when there's clouds, there'll be less photosynthesis. So we'll be able to assess the real-time carbon sequestration temporally. Now, that's the sequestration in real time. Of course, mangrove forests have emissions of carbon dioxide and methane. So we'll be measuring the methane and carbon dioxide from soils and water over low tides and high tides throughout the water column, if you look at the pictures on the left. And we'll also be measuring methane and carbon dioxide emissions from trees. And Dr. Jorge Hoyos, who will be speaking later in the session, actually kind of had this idea from 2022, when we started as part of the MRV project. We had a workshop in Jamaica, and he asked a random question with regards to the approach that was being taken, whereby we used a terrestrial laser scanner to recreate the tree in 3D. So I had just been focusing exclusively on the volume of the tree, which is to do with the amount of carbon in the tree. He asked the question, can you get the surface area of the tree? And it took me about 30 seconds of thinking, because I hadn't thought about that before, and to realize, yes, you can. So by measuring, again, a very small spatial extent of the tree, the flux from the tree, like in the picture on the right, we would be able to now have the total surface area of the tree. And so you would be able to extrapolate how much methane is being emitted by the roots and trunks. And also we'll have to see exactly where the methane peters out as you go higher up the trunk. So it's just another thing that we're going to be trying, because as far as I'm aware, it hasn't really been done, extrapolating from very small measurements to individual tree measurements, but then also to landscape measurements, because there's relationships between we'll have tree heights from the LiDAR and TandemX, as well as surface areas from the in situ measurements using the ground-based LiDAR. So we're trying to basically come up with an alternative methods to eddy flux towers, because our original idea was to put in an eddy flux tower to measure landscape level carbon dioxide and methane emissions, but cost and logistics, it was simply beyond the capability of our organization. So that's where we switched to this in situ system to try to see how we can get the same level of assessment without the high level of infrastructure and modeling required. And eddy flux requires so much resources to keep going. So that is why we're taking this different approach. And with that, I'd like to say thank you.
Muchas gracias a la doctora Juman y al señor Asmath.
Thank you very much.
Nuestra próxima ponente es la
Our
doctora
next
Hannah
speaker is
Morrissette.
Dr.
La
Hannah
doctora
Morrissette.
Hannah es bioquímica de humedales
She's
costeros del
a chemist of the
Smithsonian
Smithsonian Environment
Research Center
Research Center
y miembro del grupo
and a
técnico
member
científico
of
de la
the
Global
technical group of the
Mangrove
Global Mangrove
Alliance.
Alliance.
Asimismo, trabaja con varios países
Dr.
de América
Morrissette works with
Latina,
several
el
countries in
Caribe
Latin
y
America and the
desarrolla
Caribbean
proyectos de humedales
to
costeros
develop
que se
coastal
alineen con
wetland
metas
projects that
nacionales e
align within
internacionales
national and
y
international
política
climate policy
climática. Su
goals.
investigación se
Her
enfoca en el
research
movimiento y la
focuses on
transformación del
carbon
carbono
movement
en
and
manglares,
transformation in
marismas
mangroves,
y praderas
marshes, and
marinas.
seagrass
Damos la
beds.
bienvenida.
Let's
Su
welcome
presentación se
her.
titula Más
Her
allá del
presentation is
carbono
titled
azul,
Beyond Blue
servicios
Carbon Coastal
ecosistémicos
Ecosystem
y sus
Services and Their
impulsores en el
Drivers
Caribe.
in the Caribbean.
The format looks a little weird, so I don't know what's going to come next in the other slides, but bear with us. Hi, everyone. I am Dr. Hannah Morrissette, a coastal wetland biogeochemist at the Smithsonian Environmental Research Center. And I'm really, really happy to be here today. I work mainly in the Latin American and Caribbean region, and I'm very fortunate to consider many of you colleagues over the last few years. So thank you for continuously working with me, but also inviting me here today to talk about some of the work we've done in your countries together. I have been asked today to give a talk on ecosystem services, and I will, right? But we're going to focus a little bit more into several particulars on the drivers of these ecosystem services, because as been mentioned a couple times today already, we already know what they can do, right? And we already know what happens when they're gone. So this is from both acute and long-term losses that happen, within minutes, years, decades. But what we kind of know are the drivers to these certain ecosystem services, right? And so we rely on them, and we've relied on them forever, right? As communities, as populations, as just health and wellness of wetlands long term. But we rely on the wetlands themselves' ability to survive, thrive, and continue to provide these services over time. So what we are getting a better idea of currently is why it's like the drivers behind these services, both spatially and temporally. Yeah, a little bit off here, but that's okay. So knowing these drivers can put us in a better place for the future, right? So there's a link here. We know what the wetlands can do. We know the ecosystem services. We like the ecosystem services. We want to continue having the ecosystem services. So let's figure out how to keep access to them over time and what is actually both driving them across scales, right? For ourselves individually, on project basis, national levels, and more. And a lot of these things change based on scale, right? So if you're looking towards a more global level here, you're talking about climate change, El Nino years, thermohaline circulation. But if you move down the line here, you're talking about regional aspects. You could be looking at things like biospheres, currents, temperature changes. If you move more national, and again, I mean, national can be that could be a huge variety in itself, but now you're looking more towards watersheds and things such as, protected areas, land use, land cover changes. And then if you drive right down into the local level, you're talking about management plans, you're talking about community initiatives and that sort of thing. And we're zooming in on Belize City if you didn't know or couldn't tell. But a lot of this can come down to that coastline, right? That coastal development aspect. And something we've been working on a lot in my groups and across different countries is geomorphology. And I might test your capabilities as translators here with these next few slides. Apologies for that. But we've been working with partners across the region in a couple different countries to talk about coastal geomorphology. And this is really the study of the shape, right, and composition of our coastlines. And a lot of people have done it over time with different, with a different focus. This is one particular diagram. You can find many from a few years ago from Townsend Harris et al. 2020. But what we're looking at here is kind of the differences and the drivers behind why our coastline is shaped the way it is. So you have on the top half, you have the coastal geomorphologies that kind of recede back into like landward direction. And on the bottom here, you have ones that proceed seaward. And then as you move left, you have increasing tidal power, and move right, you have increasing wave power. And this is all having to do with hydrology, right? The movement of the water, substrates, et cetera. So the different variations of the coastal wetland ecosystems that we all work in, we all know, we all love, they live in several different types of these coastal geomorphologies. And having a mangrove focus myself, I work mainly in these estuaries, lagoons, deltas, and a lot of our open coastlines. So this slide links those geomorphologies and the effects and what affects their development on the scales that I was talking about earlier. So you're moving from large scale things, right, like climate, larger than man-made boundaries, larger than national, regional efforts like that, right? And then as you move down towards regional scale, you have more mesoscale operations. We're talking about forest level information here and getting down to the microscale processes that affect our individual sites. And a lot of us work on these very specific processes here, but we all need to link them at higher as we go towards things like policy, management, international scales, right? And in relation to mangroves specifically, many efforts have been made to classify these mangroves within this geomorphological landscape. So looking at the largest set of classifications over here, we have those four that I mentioned earlier that we typically work in, right? We have deltas, we have open coastlines or our oceanic islands, we have lagoons and estuaries. And you might have seen these maps, but Tom Worthington and the team, have been able to produce a global map of these biophysical typologies, geomorphologies, and mangroves around the world. And this is zoomed into our region of interest here. But you can see those four different types are in the different colors. You have deltas in the like dark purple, estuaries in blue, lagoons in yellow, and open coastlines in green. But the team has also classified the ecosystem setting that these categories are sitting on, right? So what type of soil, what type of substrate are we talking about, into either terrigenous, which is, land-based materials, silts, clay, and then carbonate ecosystems, which is a formulation of basically calcium carbonates and marine organisms. So you'll notice that the region is dominated by the carbonate ecosystem setting. So that's the shaded parts in pink. And we get a lot of nuances here regionally, and that's okay. Not every, global maps are never going to be 100% perfect. We know that for sure. And if you take Belize, for example, if you dig into those layers, you'll see that they classify almost the entire coastline of Belize as a lagoonal mangrove geomorphology. And while there are a lot of lagoonal ecosystems in Belize, I have many sites on riverine ecosystems, open coastlines, etc. But because of that nuance, regional and local scale mapping of these geomorphologies has also been done. And this is an example from right here in Colombia with Gustavo and team. And when you move to that smaller resolution, right? So as the ecological typology, based on that water flow and things, you can focus on the different questions that are more relevant to community levels, right? Subnational information. Because as we know, right, different types of mangroves provide different ecosystem services. And this isn't necessarily that they're discrete differences. It's more that a lot of mangroves can provide the same services, but in different levels, different capacities, different levels of importance to the communities and things that rely on them around. So we know this inherently, right? Just being in the mangroves, just studying them ourselves, just going into those forests on a day-to-day basis. We know that some areas are better for fisheries. We know those really dense intact forests are going to probably protect our coastlines more. We know that the riverine sites could have a lot of sediment delivery and therefore maybe a lot of carbon. So we inherently understand these differences, even if you're not studying specific geomorphology itself. But one place we've been highlighting these differences is in Belize, and where there are many organizations working on national level policy and management. So national environmental level management, policies such as the nationally determined contributions, or NDCs, the emissions reductions plans as part of the Paris Agreement. In Belize, they have, well, let me see if I get this right. They've just, earlier this year, they have submitted their third NDC. So their updates have now committed to the protection of 14,000 hectares and the restoration of a little more than 4,000 hectares by 2035. And this, what comes from that, right? That's a lot of mangroves. That's a lot of work. But you need to prioritize what each type of mangrove ecosystem can do for you, so that you can, the community level planning and landscape and all, to help plan out these additional protection levels, right? So zooming into that community level for local level management, we can better understand what the services are that are needed, and what we can expect to get out of that area, right? Related to the individual and the livelihoods. Because this is blue carbon and wetlands conference, I'm going to talk about carbon for just a little bit. But here in Belize, we see that these carbon dynamics vary both short and long term, right? So on the y-axis here, we have our average sediment carbon in megagrams per hectare. And then on the x-axis, we have our three different geomorphologies that I mentioned, those keys or open coasts, lagoonal and riverine ecosystems. So within each of the three, we have different boxes. These boxes are binned in the amounts of carbon per depth profile. So each color indicates a 50 centimeter segment of depth, and then it moves as it gets darker in color and it moves left to right, your depth gets deeper. So these are samples collected from, this is on a per sample basis, from over 111 plots throughout Belize. And so these are categorized into their individual depth within their individual geomorphology. So here we have the three different geomorphologies, and you'll notice a couple things right away, right? So there are several opposite trends here. In the key systems, it looks like carbon is decreasing in content as you move with depth. Lagoon seems to be the opposite, right? Seems to have more carbon as you go down in depth. And then those riverine sites seem to have a very consistent amount of carbon per depth bin as you move deeper. And our maximum here was 3 meters. Now, you'll also notice that it changes based on the depth that you're talking about, right? So if you look to the very top levels, you'll see that offshore keys have the densest amounts of carbon, then riverine, then lagoon. And this all this is driven by a lot of different things, right? So riverine systems have a lot of sediment delivery, so they tend to have higher accumulation rates, as was mentioned before, which could inherently lead to higher amounts of carbon and more consistent layers of such. With offshore keys and lagoons, you have more of like a tidal exchange, less sediment delivery. Your carbon accumulation rates could be smaller, and they could just be accumulating in those top areas, right? But another thing we did that is going to, I think, be most interesting to you MRV people is that what happens here, we can also look at the stable isotope analysis. Sorry, translators. The delta carbon, the delta 13 carbon, so the stable isotope of carbon, delta 13C, can give you an individual signature based on where that carbon may be coming from, right? The origin. And when we can do mixing models in each sample to see how much of that carbon is from different sources. And we did this with all of our samples as well. And we are able to give estimates on how much of that carbon within these individual ecosystems and individual geomorphologies are either autochthonous or originating right within that zone, mangrove originated, or allochthonous, which means the carbon is coming from a different source, either terrestrially or marine. And we've noticed that the marine ecosystems or the riverine ecosystems on average have about 40% autochthonous or mangrove originated carbon, which aligns well with international standards. That's kind of what people, or if you don't have your own data, that's kind of what they tell you to assume. But the key and lagoon sites are showing 70, 80, 90% autochthonous or mangrove originated carbon. So you're talking about this is really, really important for accurate carbon accounting. And we know that it comes down to that more local, that smaller level scale than just national, right? So if we look to this, we can also expand across a lot of other ecosystem services, right? And as the numerous ecosystem services are often more important on a day-to-day scale to our communities and local level partners, right? So we were able to analyze these things by putting together those geomorphology layers with other layers such as population densities, nighttime light densities, Fisher days per year, that sort of thing, against those geomorphologies globally. And we have our mitigation, right, our carbon sequestration and such. But when it comes to those other things, such as maybe coastal protection, we found that estuaries and lagoons are better are protecting more property value globally on average for commercial and residential sequentially there. And deltaic systems have significantly higher value for fisheries, for finfish fisheries, and the highest overall population increases over time. So you can imagine there's going to be in the future even more reliance on these fisheries and ecosystem services moving forward. And you move to lagoonal, and they're higher value for other things such as bivalves and crabs and such. So now you're looking, and they're more residential, so you're looking towards more local, like smaller scale livelihood services, such as harvesting. So as you're moving towards these perspectives, right, we're calling for this to not solve all the problems, because I just said you can't do it globally, but to tell people to look at a more local level for this when they're putting in their management and protection plans. And that's because many countries have dedicated time and energy to these extensive additional protection and conservation measures, right, for mangroves and other wetlands. But the analyses such as these can look beyond approaches, just like drawing a box and saying this is a new protected area, but rather because we need to protect X amount of hectares of mangroves, but doing it with intention so that we can look to the future for what's both going to be helpful to the community, but also what is feasible for that ecosystem to provide. And there are a lot of dynamics that I've just mentioned, and we're not the first to do this, but it is very complicated and sometimes very expensive. But when you're looking at the local level, I mean, that's necessary. And I think we all know that working with communities and individual partners within our zones. So yeah, the analyses mentioned here today will be, submitted towards academic journals, but the data is already in the hands of our partners in the countries in which I've been invited to work. And this local level information is being put towards not only those like national level policies such as NDCs, but also localized management for individual protected areas and stuff. So if you have any input, any thoughts, any questions, please feel free to reach out.
Bueno, muchísimas gracias a la doctora
Okay,
Morrissette.
thank you very much to Dr. Morrissette. Up next, we're going to have the opportunity of having the presentation of Dr. Diana Isabel Gómez, that is the Chief of the Organization Land and Dynamics of Ecosystems for the Program of Biodiversity in Maritime Ecosystems of INVEMAR. Dr. Gómez, since 2007, is leading the research in seagrass and INVEMAR, and their experience covers ecology and monitoring of mangroves, coral reefs, and since 2016, she's working in blue carbon and seagrass, has published more than 30 different articles, and is also a key member of the Caribbean network of the Global Monitoring Network for Coral Reefs. The presentation has the title of The Ecological Value of Seagrasses in Carbon Capture and Advances in Colombia.
Le damos la bienvenida.
Bueno, muy buenas tardes.
Okay, good afternoon now. I'll try to be brief and very precise in relations to this talk because I think that you may be hungry by now. So let's get started. So seagrasses in Colombia have different species, 6 to be exact, and between these 6 species, this is one of the images. And depending on the size of what they represent in the seagrasses and the coast and also in complexity, and it comes from species that are not so prolific to the high salinity to the ones that in complexities to from the needs of the substrates. The first one will be Salophylla bailoni. The sapiens in writing is more eager to live in these fine substrates than these other two that are in the red circle that are more akin to coarser substrates. Okay, the seagrass meadows in Colombia are intermittently throughout the coastline in the Colombian Caribbean. Only that, the Colombian Caribbean. More than 80% of these are present at La Guajiras. And the 6 species that we saw in the previous slide are now here in this department. Many of the morphological and physiological adaptations of these seagrasses are the ones that are bringing us here today. Well, in this space, it is basically the structural design that consists in these rhizomes and roots that have the leaves, and this structural nodule that you see is the one that is multiplied in every direction, then creating this interlinking of these seagrasses. In general, this is the kind of reproduction by cloning, asexually. In these same waters, they also have flowers that are female and male, and these are apparently pollinized by polychaetes and other crustaceans that coming from the investigation of Dr. Van and other collaborators in 2016. So, and we talk about the fruits, and especially for Thalassotus surinam, this is one of the topics that has slowed the the studies that have to do with restoration, because it was quite hard to identify the moment in which we can find them, and then the usage of them in topics that have to do with repopulation. And also they have maritime dispersion and have epidermal chloroplasts that facilitate the photosynthesis process at any point in any place in which they are. Okay, practically the ecosystemic service of these specific ecosystems have to do that through their leaf, they dissipate the energy of the currents that flow through them, turning them in a kind of traps that are sediments that are in the bottom of these seagrass meadows. So then these, sorry? This thing had an animation, sorry about that. So this characteristic means that through this intricate root system, they are allowed to hold sediment in a way that reduces erosion. And let's say, through collecting this, they have the opportunity of then growing. They grow, and through that growth, they are creating these terraces in a way that after they are the ones that start modifying the morphology of the coast littorals and then facilitating them, then the currents that flow through them, reducing the flow or their speed, then forming a kind of wall against erosion. Also, them are considered as areas of high biodiversity due to the richness of life forms and through fishes, invertebrates, and mollusks that are living there, or that are also part of the synergy chain between mangroves, beaches, and these seagrass meadows and the coral reefs, and whenever they are in the same area. Well, the main carbon, as Dr. Morris was saying in the previous talk, is that the main carbon they gather is their own, and it's when their leaves, when they die, it is there in the same sea meadow, in the same kind of sediment, unless they have high tide that moves them to other places that are nearby, maybe the beach, maybe other places in these soil, or even through the deeper soil. So these ecosystemic services are, you need to have some criteria, or you can see them here. And the characteristics of the physical environment are going to determine the degree of things that they're adding as an ecosystemic service that we might want. The dominant species are also going to have some influence on that. It's not the same thing to have these meadows of Salophyllus that are 3 centimeters, or Thalassotes urinosa and Ingodium for these cases. Out of conformation and the extent of this meadow is also going to be a preeminent factor in the way in which this is going to provide this service, the age of the plants as well. It is not the same thing to have a meadow that maybe tens or even thousands of years with big terraces than one that is recently restored and that is not yet at the point of achieving the potential that we expect from it. The time experience present in the science, the tensors that are present, and also the associated ecosystems. So whenever we request enough on maritime ecosystems, for example, these meadows, when we say that there are going to be solutions of center kind of deficits that we need from the point of view of what we do, humans, we need to bear in mind that any kind of activity of repopulation is going to take time, or it's going to be a process of trying to establish these spaces and time and the proper characteristics that are required for the work that is being requested of them. One of those is especially the thing that we talk about, the terraces. And in this exercise, you see in these pictures, these are some of the images that are seen in the Blue Carbon Initiative document. This was one of these terraces or Posidonia oceanica, and it's the whole terraces, and this is a small one that is It is around 40 centimeters, and this was taken here in the Tayrona Park. In the archipelago of San Andres and Providencia, we have these torsos that are a meter deep as well. So based on these characteristics is that we trust or that we have the hope for the potential of these seagrass meadows. So in the framework of the project in mangroves, seabeds, and local communities, the development of exchange of experiences for in the comprehensive management of biodiversity and its services in the Caribbean region that was developed between 2017 and 2020, we did a small, well, maybe a small project within the project to be able to determine what is the role of these sea grass, especially the Thalassia tesudinum, in the context of the Caribbean. So the area of study was mainly the department of La Guajira, the ones that you see here as A, the north part, and area D, that is San Andres Island, and these other intermediate points that are as reference, one in the Cartagena Bay and also in the National Park Tayrona, and each one can with different characteristics. Alta and Media Guajira is very sandy within the surface rubble, and the wind of the tide is due to the wind and has low anthropogenic disturbances. The Natural Park Tayrona, the place in which we work in Vallatengue, it is a bay that is very quiet and calm, part of the continental platform, shading coraline, and also associated with mangroves and corals, and again, with relatively low anthropogenic disturbances. The Cartagena Bay also is the area that we see that is within a protected bay, but even though that's the case, this is an area that's highly industrialized and has suffered a loss of seagrass meadows in the last century of around more than 80%. And the area of San Andres that are associated to this reef lagoon and are surrounded by mangroves and the coraline environment and microalgae, and has medium anthropogenic disturbances due to tourism. We use the methodology for the Blue Carbon Initiative. We have 14 sites across different localities that were sampled. We have 65 cores, and they were corrected for compression. Only 5 samples were successful for radiocarbon method and 6 for plum 210. And these carbon-14 analyses were made in the United States in the AMC Director Laboratory, and the organic matter was made in IMEMAR. And with the University of ECU in Australia, we have information of these carbon isotopes and numerical analysis. And the results we see that for these projects is that the average of the carbon stock at a meter depth was 241 milligrams carbon by hectare, and this is higher than the ones you see in Mexico, United States, and Indonesia that was around 71 and 170. And the global estimates from is 140 milligrams of carbon by hectare. And in the same way, we have the accumulation rates of sediment, and you see that are higher than the range that was on the papers at that time, and this was made by DC Soster Marina Meadows, and the same is true for the rate of carbon soil. It is also higher than the Dutch Caribbean, also Australia, and you see that the information of 14 teragrams of carbon of organic carbon in the first meter of soil, so that is 52 teragrams of equivalent carbon, is around 64% of the emissions from the fossil fuel burnings in Colombia in 2014. And based on the rate of accumulation of carbon, we notice that 0.3 teragrams of carbon a year is more or less 0.4 of the emissions of fossil fuels in Colombia in 2014 rates. And this has given us a lot of energy and a lot of optimism to think that we have a good hotspot in seagrass meadows in Colombia. To the isotope analysis, when we analyzed the origin of the carbon that was shown in these samples, what we noticed is that in general, the nitrates of these seagrass and mangroves were the main components in all the sediments that we analyzed. Ceston, especially for the place of Media Guajira, was quite relevant. As you can see, for San Andres, added from Ceston and seagrass, we also see have these Alameda tuna. There is also very important, as you notice, and mangroves were relevant enough in the areas in which they are more prevalent, as for example, the high Alta Guajira, Tayrona, and Cartagena Bay. One of the things that was identified is that the apparent density of the sediments showed that was around 0.016 of organic matter by percentage, or 0.12 of organic carbon, and around 0.9 milligrams of carbon by cubic centimeter. So the apparent density was increased with the depth, and organic matter and organic carbon decreased with the depth in all these sites. So the relevant thing about this is that whenever they did the analysis for the granulometry of this, in the sites in which they found higher stocks of carbon, you saw that the amount of particles were lower than 0.016, so between 0.016 and 0.125. That was the main constituent of this material, and the coarse sediment were only identified in Cartagena, in the Bay of Cartagena, and it was not relatively, well, not that representative here. This one also had an animation, and what I wanted to show here or mention here is that this first exercise with the MapCo method was done to the higher part of the La Guajira department, from a point that is up north, and also to Bayandita, that is, and you cannot see that on the map, but this is between 2017 and 2020, and after that we did another exercise, and in the middle part of the department of La Guajira, that is the one that I'm talking about, that has more than 80% of these seagrass meadows to the south. And in these red spots, we see the recovery of information for, by the course of these Thalassete studinum and Silentiliforme, just to take a look at the differences that might be present. And from those differences, we noticed that the amount of these fine sediments that we needed, or that we identified in the previous study, and that were corresponding to the higher rate of carbon stocks, were not found in this other exercise, or this in this document that we are working on. We noticed that the carbon stocks are between 18.8 and 45.3 micrograms of carbon by hectare. So this means that there are some differences that we need to consider. And other thing that we also need to consider is going to be biomass. We did this exercise with live biomass in total in the department of La Guajira and also in San Andres. So this exercise is showing that even though San Andres that had only 3 stations, in La Guajira had 8, it was more or less at the same level that it was obtained on La Guajira. Only the data of air biomass was more representative in one of the sites on La Guajira. It was in Carrizal, and the average of underground biomass and the total average was, you can see, was San Andres. So in general, the biomass average for San Andres was 4.6 versus 3.5 for La Guajira, that had more stations as the one that has more or the higher amount of seagrass in Colombia. So the global average was 2.5, and then based on this exercise, we are still above the average. If we maybe compare ourselves with other exercises that were done in other areas of the world, if we could compare to the first one that we did with the MAPCO, we would be located in the with sediment, and the amount of carbon stock in sediments, we would be between the Oriental Atlantic and Australia, and then biomass we would be between Australia and the Mediterranean. But with this last exercise that we did, it would be between North Pacific and Indian Pacific. So this shows that apparently the carbon stocks are associated to the fine sediments, because these are the ones that actually avoid or stop the remineralization of these elements between these sediments. In the areas in which these sediments were fine or less coarse, you see the comparison here. The average was around 241 milligrams of carbon per hectare. And the ones in which we was not as fine, it was between 18.8 and 45.3. So the total for the biomass of the data show, we saw that the contribution to the organic carbon conservation in general should not be underestimated, even though it is lower than the one recorded for the sediments. But we also need to consider that the aerial biomass is maintained, the carbon and on the sediments is also going to be maintained throughout time. Many specific studies need to happen in things to do with methodology, because every single time that you have a solution for one of the questions, or you are going to have other 3 or 4 questions that also need solutions, or need other studies, or need other things to happen to have a certainty of these results. This is basically what I wanted to share. So I don't know if we have time for any question. No? Okay. Okay. Thank you very much, Joanna. And yeah, that's true. We don't have time for questions and not for the discussion panel, precisely because we are short on time. We need to follow this schedule. But to finalize with this technical block, we invite all the speakers that presented today to do a group picture here in this stage. And we also invite the moderators for them to get close. The technical team wants to have a small meeting with you guys before having our lunch. For the rest of the attendees, we recommend you to start moving. We are now going to design area for the lunch. Our volunteers are willing to guide you if you maybe need that. And remember that Inbemar is a space that's free of smoke. So we remind you of that, and we thank you for your cooperation. We also remind you that this event is being photographed with promotional, and so if you haven't been there in the selfie booth, it's outside. If you want to upload your photos to social media, please tag us as @BlueCarbonMBR. And also use if you maybe need to take a look at the updated agenda, is in the Huba app. If you haven't fill the form for the event of the field trip, is just for 50 people. So if you want to go there, please do. It's going to be a good field trip to the Sierra Grande de Santa Marta. So please do if you have done so. We are going to get back to this auditorium at 2 p.m. so we can continue with the conference. Please enjoy your lunch.